So, our founder of Amber's Academy is also here with us. Assalamu alaikum.
वालेकुम सलाम। जी सिजा, कैसी हैं आप?
Yeah, I'm fine. We took a namaz break for 10 minutes. So, I believe
ओके।
I have already texted on the group that the students can join. The classroom is open, but you can begin with your introduction.
ओके ओके ओके, बिस्मिल्लाहिर्रहमानिर्रहीम, अस्सलाम वालेकुम। मेरा नाम अमरीन रशीद खान है एंड मेरी एसोसिएशन अंबर अकेडमी से बहुत पुरानी है, बल्कि मेरी ही नहीं, मेरे पेरेंट्स की भी। वी स्टार्टेड दिस जर्नी इन मिड ऑफ 1980s। मेरे पेरेंट्स ने शुरू किया ये मेरे ही नाम से अमरीन कोचिंग सेंटर इन 1985। और उसके बाद मेरे पेरेंट्स ने इसको रन किया फॉर क्वाइट 10-12 years या उससे भी ज्यादा। फिर उसके बाद मैंने इसको जो है वो तकरीबन 10 years रन किया। और अब हमारी थर्ड जनरेशन शिजा इसको रन कर रही है। तो वी हैव अ लेगेसी ऑफ टीचिंग, एकेडमिक एसोसिएशन। मेरे पेरेंट्स भी टीचर थे, मेरा ताल्लुक भी टीचिंग से है। आई एम द असिस्टेंट प्रोफेसर इन वन यूनिवर्सिटी। और शिजा भी डॉक्टर बनने के साथ साथ जो है वो उनका भी पैशन जो है वो टीचिंग है। तो टीचिंग हमारे पैशन के थ्रू हम करते हैं। और आप लोगों से भी ये रिक्वेस्ट है कि इन सेशंस को सीरियस लें। अपार्ट फ्रॉम के ये अगर फ्री ऑफ कॉस्ट है तो हमने लेना है या नहीं लेना, अपना टाइम को स्केड्यूल करें। जो आपका टाइम है उसको कोशिश करें कि लाइव क्लासेस लें। और कोशिश करें कि यहाँ पर जो आपको टास्क मिलते हैं और उसको कंप्लीट करें, क्योंकि अगर आपने अच्छी ग्रेड्स लेने हैं तो आपको शिजा एक तरफ सब पढ़ाएंगी, लेकिन इससे अलग आपको खुद को पढ़ना भी पड़ेगा। ठीक है, तो ये दो दो लोग होते हैं जो साथ काम करते हैं टूवर्ड्स अचीवमेंट ऑफ द गोल। और गोल क्या है कि हमारे ग्रेड्स अच्छे आए, एक पढ़ने वाला और दूसरा पढ़ाने वाला। अब पढ़ने वाला जितना भी आपको, पढ़ाने वाला सॉरी, कितनी मेहनत कर लेगा, और अगर पढ़ने वाला सीरियस नहीं होगा उसको लेने में, वो अलाइन नहीं होगा, तो वो ऑफ कोर्स वो रिजल्ट नहीं आ सकता। तो सीरियसनेस बहुत जरूरी है, कमिटमेंट बहुत जरूरी है, डिटर्मिनेशन बहुत जरूरी है, नॉट ओनली फॉर बायोलॉजी, फॉर ऑल द सब्जेक्ट यू नीड टू बी वेरी फोकस्ड, वेरी सीरियस इन दीज। ये जो ओ लेवल्स के हमारे साल होते हैं, क्योंकि यहीं से हमारा करियर जो होता है वो डिफाइन हो रहा होता है कि आगे हमने कहाँ जाना है। तो ये छोटा सा इंट्रोडक्शन था मेरा। आई एम डूइंग PhD फ्रॉम मलेशिया इन मैनेजमेंट, and by profession I am a computer system engineer. And then I did my MBA from IIM Karachi. Basically, I'm from Karachi, was born and raised in Karachi, and then recently we moved to Lahore just a couple of years ago. So we are now living in Lahore, but keep on switching between two cities in Lahore and Karachi because I have my own home at Karachi. So hopefully you will have a very interesting and informative journey with Shiza Amber, with your of course subject is biology. उम्मीद है कि आपका ये सफर बहुत अच्छा होगा और रहेगा शिजा के साथ। इट्स अ लॉन्ग टाइम कमिटमेंट, इट्स अ कमिटमेंट ऑफ आई थिंक ओवर फॉर द कंप्लीट ईयर के लिए है। तो इसमें जितना आप सीरियस होंगे, जितना फोकस होंगे, जितना आप अपना एफर्ट पुट इन करेंगे, उतने ही आपके ग्रेड्स अच्छे आएंगे। क्वेश्चंस को पिक करना, उसकी टैक्टिक्स को समझना, एक क्वेश्चन को कितने कितने तरह से जो है वो आप लोगों का आता है ट्विस्ट एंड टर्न होके, उन ट्विस्ट को एक्चुअली अंडरस्टैंड करना, ये सब चीजें बहुत जरूरी होती हैं। ए लेवल्स में भी और ओ लेवल्स में भी। ए लेवल्स में और डिफिकल्टी लेवल आपकी इंक्रीज हो जाती है मैनी फोल्ड्स। और ए लेवल में अगर आप बायोलॉजी स्ट्रीम में हैं और इन केस इफ यू आर हेडिंग टूवर्ड्स फॉर द मेडिकल स्ट्रीम, तो फिर तो आप सोच लें कि आपने कितनी ज्यादा इसमें एफर्ट पुट इन करनी है, क्योंकि जो आपका कंपटीशन है बेसिकली वो होता है इंटर स्ट्रीम के साथ। एंड दे हैव वेरी गुड गुड रेटाफिकेशन, इनके मार्क्स आपके जाते हैं 90+, 85+। तो फिर वो मेडिकल में उनसे कॉम्पीट करना बहुत मुश्किल हो जाता है, क्योंकि ए लेवल्स में मार्क्स स्कोर करना बहुत ज्यादा चैलेंजिंग होता है। तो वंस अगेन फिर उसकी इक्विवेलेंसी बनती है और वो सारे चैलेंजेस होते हैं। तो उन चैलेंजेस से जैसे कहते हैं कोप अप करने के लिए बहुत जरूरी है कि जो आपका ये 1 ईयर है इसमें आपकी इतनी स्ट्रांग ग्रिप हो एएस में भी और फिर A2 में भी, ताकि अगर कहीं ना कहीं जो है वो आपका कोई ऐसा आपका जो है वो वीक पॉइंट ना रह जाए जिसकी वजह से आपका स्कोर कम हो जाए। और स्कोर कम होना ही नहीं चाहिए, बिकॉज मैंने आपको बताया कि आपकी जो मेडिकल स्ट्रीम है वो बहुत ज्यादा टफ कंपटीशन देती है। शिजा को उसका बहुत अच्छी तरह से आइडिया है। तो विश यू ऑल द बेस्ट फॉर योर कंप्लीट जर्नी विद शिजा अम्बर। और आई होप एंड प्रे फॉर ऑल ऑफ यू कि आप लोग अपने करियर के गोल्स अचीव करें। और once again I request for seriousness and commitment. So that's all from my side, Shiza.
ठीक है, Jazakallah khair and Jazakallah for joining us. So we'll formally begin our session. So the first topic is quite easy. It's of cells, and before starting, before going ahead, I would like to mention one thing again that I'll be here throughout, inshallah, and I'll be with you in contact. You have my number, you are added into the WhatsApp group. If you face any difficulty anywhere, you just drop me a message and I'll try to respond as soon as possible. Because the reason why I'm doing this is because when I was doing my A levels and when I was caught up in the past papers, in the preparation and the tough concepts, and when I used to message my teachers, we did not have that much time to respond, and many of my messages were just left on scene, and I never got reply from my teachers. And so this is I'm not taking this towards a negative side. I'm just trying to help you out, and I'm just trying to communicate the fact that when you get into your A levels and when you'll be caught up with so much of the stuff, then I'll be there for you, and I'll try to help you out in the best way that is possible. So without any further ado, we are beginning our first chapter, and it's quite easier one, but of course we have much more concepts in A levels, like I said. So we are starting with cells and okay. So cells, we have two basic categories of cells. Okay. And I'm not talking about animal cell and I'm not talking about plant cell because this was the difference that you learned in your O levels, right? But I'm formally talking about the main difference between two types of cells, and those two types of cells are prokaryotes and eukaryotes. Okay, I'm not mentioning animal cell and plant cells. Okay, so there are basically two categories of cells. One of them is prokaryotes and other one is eukaryotes. Okay, so prokaryotes. Let's just break this word into two. We need to see that there is a word pro and there's a word karyot. Okay, and then here we have this word u and again a karyot. So pro means not true. Okay, and u means true. So pro is not true and u is true. While karyot means nucleus. So karyot in both of the scenarios will mean nucleus. So we have a not true nucleus and we have a true nucleus. The one with true nucleus is known as eukaryote, and the one that is known as not true nucleus is known as prokaryote. So the basic difference between prokaryote and eukaryote is of nucleus. That eukaryotic cells will always have a proper nucleus located in the center, like you learned in your O levels that there is a nucleus in an animal cell and in the plant cell, right? But in prokaryotes, we do not have a nucleus. So basically, you need to break down this word prokaryote and eukaryote, and you need to remember the difference between both of them because this is a very repeated questions in your paper twos. So again, prokaryote literally means not true nucleus, and eukaryote means a true nucleus. Okay. So acha ji. So the example of eukaryotic cells are plant cell and animal cell. The way you have been learning about plant cell and animal cell, that both of those cells are going to come under the category of eukaryotic cells, while prokaryotic cells are the bacterial cells. Okay, so this is the example, and we just literally discovered the meaning of prokaryote and eukaryote. Now we need to dive more into the differences of prokaryotes and eukaryotes. So during your O levels, did you guys learn about ribosomes? Are you aware of ribosomes? small dots present in the cells, or this is a completely new concept for you? I believe you learned about cell membrane, you learned about cell wall, you learned about nucleus, you learned about vacuoles. Did you learn about mitochondria? I'm not asking that do you know a deep information about mitochondria or any of the organelles. I'm just asking that are you aware of their names?
Yes, yes.
Okay. What about you, Abdul Rahman?
Yes, but not ribosome.
ठीक है सही है perfect कोई बात नहीं अच्छा जी so we are all aware of the fact that cells do have organelles right हर सेल के अंदर organelles होते हैं ना we have a
Yes,
nucleus we have chloroplast we have vacuoles we have cell membrane we have cytoplasm ठीक है these were the basic organelles okay so now an additional organelle that we have in cells is of ribosomes ठीक है so ribosomes are literally dotted structures like this. We'll go through the diagram as well, but I'm just trying to explain you one thing, and we'll go deep into the function of ribosomes as well. So ribosomes are literally like these dots, okay? And basically, we have two subunits of ribosomes. So ribosomes will appear like this with two subunits. सही है अभी we are not going to dive into much detail for ribosomes because ribosome का पूरा एक अलग chapter है of nucleic acids. So we'll discover the structure properly into that chapter. But अभी के लिए what we need to understand is that ribosomes are dotted structures with two small subunits. Make sense? Are we all clear? All right. So we have basically two types of ribosomes. We have two types of cells, right? We have prokaryotes and eukaryotes. Similarly, we have two types of ribosomes. One of them, and the difference between them is just of size. So one of them is 80S and one of them is of 70S. And what we mean by 70S, so S is basically the unit that we'll measure ribosomes in. For example, for length, we have meters. For weight, we have, for mass, sorry, we have kilograms. For weight, we have newtons, right? So we have unit for every physical quantity. Similarly, for ribosomes, we have two sizes, 80S and 70S. Make sense? Are we all clear? Perfect. So ribosomes are dotted structures with two small subunits, and there are two types of ribosomes. One of them is 70S and another one is 80S. So 70S ribosomes are a bit smaller than 80S ribosomes. Okay? And 80S ribosomes are always found in eukaryotes. And what were eukaryotes? A quick recap of eukaryotes that eukaryotic cells are those cells which have a true nucleus, like we studied in O levels. And the examples that we just covered for eukaryotes are animal cell and plant cell. Okay, they have a centrally located nucleus. Perfect. Are we all clear? So eukaryotes have 80S ribosomes and prokaryotes have 70S ribosomes. Same. Then moving towards the cell wall. So we do not have a cell wall in animal cells, right? We have a cell wall in plant cells, and that cell wall is made up of. Quick check. Cell wall is made up of. Oh, cellulose. So cell wall in plant cell is made up of cellulose. Okay. And we have one more eukaryotic cell of fungi. So fungi has a cell wall made up of chitin. And you need to remember these components that make up the cell wall of prokaryotes and eukaryotes. So we are talking about eukaryotes right now. That plant cell has a cell wall made up of cellulose, while fungi has a cell wall made up of chitin. Then, other than that, we have bacterial cells that come under the category of prokaryotes, like we discussed. They do not have a true nucleus. So their cell wall is made up of peptidoglycan. Okay? Make sense? Their cell wall is made up of peptidoglycan. And peptido literally means peptides, proteins, and glycan means carbohydrates. So the cell wall of bacteria is made up of proteins and carbohydrates, and collectively we call it peptidoglycan. So one thing that it is not mentioned here, but we need to remember that the other component that makes up the cell wall of bacteria is murin. You need to write it as well. If you are making your notes, you will get the recordings and the slides as well, but try to make your own notes as well. So murin is also the component which makes the cell wall of bacteria. Perfect. Are we all clear? Do we have any question? Abdul Rahman?
No.
No. Okay. I believe we have, I think Iman just joined us and Hira. Iman, just a quick check that can you unmute yourself and introduce a bit about yourself, because we just went through the student intro and the intro of mine and the intro of the founder of Amber's Academy. Iman, can you unmute yourself if you are comfortable, or you can just type in the chat box. Cellulose. I'm Iman. Okay. So Iman, you are from Karachi, or um, and I have big biology as my subject. ठीक है. And you are beginning your A levels from? Are you a private student or are you enrolled in some college? Meanwhile, from Beacon House. Oh, great. I'm also a Beaconite. I completed my AS level from Beacon House, and I did my O levels from Beacon House as well. So technically, you are my junior, like Abdul Rahman. Abdul Rahman, you are my junior. Oh, my mother knows you actually. All right. ठीक है जी. Hira, would you like to introduce yourself? Would you like to unmute yourself, or um, would you like to write your intro in the chat box? Because this is the first session, right? I need to know all of you. I need to know whom am I teaching. So I'll not ask about your student intro in the upcoming classes, of course. Miss, I'm Hira. I also belong from Karachi. All right, Hira, and you're beginning your A levels from college, or are you a private student? Private. Okay, perfect. Doing A levels as a private student is way more better than doing A levels as a college student. I'm telling you from my personal experience. It was quite tough to carry on my A levels from Beacon House, like I already mentioned. So, okay, a quick recap that we have begun the first chapter of AS levels. You can also go through the recordings of what we just discussed. We just discussed about our intro. We discussed the strategy that we'll opt for this academic year, and now we are beginning with our first chapter, that is of cells. We briefly discussed that there are two types of cells, and I'm not talking about plant cell and animal cell. I'm talking about the categories of cell. The categories of cells are prokaryotes and eukaryotes, and we broke this word of eukaryotes into two, and we also similarly broke down the word of prokaryotes into two. And pro literally means not true, karyot means nucleus. So prokaryotes are those cells that do not have a nucleus. And the main example of prokaryotic cell is of bacteria. Then eukaryotes, they have a true nucleus, centrally located nucleus, like we already covered in our O levels. We talked about plant cell and animal cell, so they come under the category of eukaryotes. This is exactly what we have discussed so far. And then I was talking about this new organelle. This new organelle is ribosomes. You do not need to worry if you are unable to understand ribosomes right now, because we'll extensively discuss ribosomes in chapter number 5 and in this chapter as well, but later on. So I was just discussing the structure of ribosomes, that it appears like a dot and it has two small subunits. And there are two types of ribosomes, like two types of cells. So we have two types of ribosomes, 70S and 80S. So the basic difference between these two types of ribosomes is of size, that 70S ribosomes are smaller, 80S ribosomes are minutely, they are bigger. Okay. So, and then we were discussing that eukaryotes have cell wall and only plant cells have cell wall. So their cell wall is made up of cellulose, while fungi has a cell wall made up of chitin. And these are very important. You need to remember that cellulose is made, plant cell wall is made up of cellulose, and fungi cell wall is made up of chitin. Then we were talking that prokaryotes have a cell wall made up of peptidoglycan, and the other word is murin. So peptidoglycan, if we break this word, so we'll discover that peptido is proteinish part, and glycan is the carbohydrate part. Okay. And the other name for peptidoglycan is murin. So you need to remember these words, because when we'll do the past papers, these words are going to come again and again. Then we discussed the example that bacteria are prokaryotic cells and plant cell, animal cell, fungi cell, they are eukaryotic cells. So eukaryotic cells, once again, they have a true centrally located nucleus. Then we were talking that prokaryotes, they have additional features, and these features are not present in eukaryotic cells. These features are capsule, pili, inner foldings, flagella, and plasmid. So you'll have to remember these. We also see how they appear and what are their functions, but up till now, you need to remember that they have those features that are not present in eukaryotic cells. We have never seen a capsule, we have never seen a pili, we have never seen an inner folding, flagella, or plasmid in an animal cell, right? Then coming towards their division, so eukaryotic cells divide by mitosis or meiosis, and we'll talk about mitosis in chapter 4 extensively. Meiosis, will cover meiosis in 8 levels. So briefly, you need to remember that eukaryotic cells divide by mitosis or meiosis. Then prokaryotes divide by binary fission. This is an important thing you'll have to remember. Okay, so whatever I highlight or whatever I underline, you need to remember these things, because this is exactly what you will have to write in your past paper, in your paper, sorry. ठीक है. So ठीक है. Mitosis and meiosis is a way of reproduction in eukaryotes, and binary fission is a way of reproduction in prokaryotes. Then in prokaryotes, because prokaryote bacterial cells, they are very simpler cells, so they do not have any membrane bound organelles like nucleus. They do not have a nucleus, we already talked about this. They do not have ER. So ER is endoplasmic reticulum. We'll talk about this as well. Just keep this in mind that prokaryotic cells do not have any major organelle like nucleus, ER, Golgi body, or mitochondria. While eukaryotes have all the membrane-bound organelles, they have chloroplast, they have nucleus, they have vacuoles, they have mitochondria. Then you need to remember that prokaryotes have a circular DNA and they do not have any histone proteins. So have you seen the structure of DNA? It looks something like this when it is condensed, right? It looks like this. So histone proteins, in order to remember what histone protein is, I'm giving you an example. Do not write this anywhere. So histone proteins are literally the hanger of the linear DNA. So this is a linear DNA, and this is usually present in eukaryotic cell, while a circular DNA looks like this. So it does not have any histone proteins. It is not in a hanging position, but your linear DNA is in a hanging position. So the hanger of linear DNA is histone proteins. Make sense? While the circular DNA, it does not have histone proteins, so this is why it is circular, and it does not appear like a linear DNA. Are we all clear? Have all of you understood the differences between prokaryotes and eukaryotes? Any question? Right. ठीक है जी. Okay. So this is a prokaryotic cell, and the example of prokaryotic cell is a bacterial cell, right? And we just talked about the cell wall. So cell wall of bacterial cell is made up of? We just went through
Urine.
it. Murin?
Urine.
Yes.
Yes.
And what was the other name of murin?
मैम याद नहीं वाला.
कोई बात नहीं. Good effort. फिर भी बहुत अच्छी effort है. Okay, so we have someone in chat. Peptidoglycan. Perfect. So cell wall of bacterial cell is made up of peptidoglycan and murin. ठीक है जी. Mashallah, Hira and Abdul Rahman. Okay. Then we were talking about the additional features that are present in bacterial cell. So these are capsule. Oh, okay. So capsule. Have you seen the medical drug of capsule? It looks like a covering, and inside it is the medicine, right? So capsule is basically the covering of the bacterial cell. It does not have any function other than the protection of the bacterial cell. So it basically encapsulates the entire cell just to protect it. ठीक है. And inside it we have cell wall. Then we have ribosomes. I just mentioned that they are the dotted structures and they have two small subunits. So we cannot see the small subunits over here because obviously they are very small and they can only be seen under the section of advanced electron microscope. We cannot see the small subunits in light microscope. We'll talk about that as well. So ribosomes, and what was the size of ribosomes in prokaryotes?
70 S ribosome.
70, yes, perfect. ठीक है जी. Then we've talked about the nucleus. So it does not have any proper nucleus, right? Because the nucleus in animal cell looks like this. It has a proper membrane covering known as nuclear envelope. But here it does not having any nuclear membrane. It just has this circular DNA with no histone proteins. And circular DNA जो है वो इस तरीके से appear होता है because it does not have any hanger like the histone proteins. ठीक है? Then plasma membrane. So plasma membrane in A levels, especially you'll say that cell membrane is known as cell surface membrane or plasma membrane. You will not be writing cell membrane anymore. Keep this thing in mind. Make a small note that you will never use this word again in your A levels. You'll write plasma membrane or you'll write cell surface membrane. If you'll write cell membrane, then you'll not get marks. You'll get a 0. Okay. So plasma membrane is the new name for cell surface membrane or cell membrane. We talked about plasma membrane. We have talked about capsule. We have talked about cell wall. We talked about ribosome. We have 70S ribosome. We talked about DNA, circular DNA with no histone proteins. Now this plasmid. So plasmid over here. I believe that you should be aware of this word plasmid because in your O level, sorry, while doing the genetic engineering topic, you must have heard about this word plasmid. No? Do you remember this word? नहीं भी आता तो कोई बात नहीं, डांटूंगी नहीं. No, I don't remember. 70 years. ठीक है जी, कोई बात नहीं. So plasmid is basically it basically contains genetic material. Other than DNA, plasmid is a small section and it also contains genetic material of the bacteria. ठीक है? Make sense? छोटा सा होता है and we usually extract it out while we are doing genetic engineering, where we combine DNA of two different organisms. We combine plasmid and we combine the DNA of human beings and we genetically engineer insulin. You'll talk about this in your A2 levels as well. So you do not need to worry if you are not aware about the genetic engineering so far. But keep this thing in mind that plasmid is only found in prokaryotic cells and it has the genetic material which we can extract in order to genetically engineer any new product. Okay. Then we have pili. So pili are these attachments. Acha. So you need to remember the difference between pili and flagellum in case if it comes for labeling, because labeling is also an essential part of AS level papers. So flagellum is just one. Okay? And flagellum is a long thread-like structure. See, this is a long thread-like structure, right? And pili, these are small thread-like structures. So pili basically what their function is to attach bacterial cell with any eukaryotic cell in order to cause the infection. Okay? So pili are the site of attachments, while flagellum enables the movement of bacterial cell. Are we clear with this difference? So flagellum is a long thread-like structure which enables the movement of bacterial cell, while pili, it causes the attachment of bacterial cell with a eukaryotic cell, mainly human cell, because infections are usually caused by bacteria. Make sense? Are we all clear? Okay. सब thumbs up कर दे because मैं समझ नहीं आता कि किसको समझ आ गया किसको नहीं आया. उस्मान एंड हमारी जो 2 गर्ल्स हैं यहाँ पर, मुश्किल तो नहीं है अभी तक? आ रहे ना समझ में? ठीक है. So we have 5 more minutes before we end this class. Let's talk about eukaryotic cell. इसको देख के घबराना नहीं है. It is the same cell that you discovered in your O levels, but it has slightly more additions, obviously, because now you are in an advanced level, you are in A levels. So we'll talk about more organelles, right? We said that eukaryotes, they have a proper nucleus, right? So we can locate this proper nucleus in the center. Like I mentioned that eukaryotic cells will always have a centrally located nucleus. Okay, we can locate this. But a centrally located nucleus was not present in bacterial cell, right? There was just a circular DNA. And in nucleus, you can find linear DNA with histone proteins. So histone proteins were basically the hangers of linear DNA. है ना? ऐसे ही था ना? ठीक है. ठीक है. So, okay, then in nucleus, we have a spherical body known as nucleoli or nucleolus. It is the site responsible for protein manufacture. We'll talk about the function of each of the organelles in the later slides, but first we are just understanding the labeling of eukaryotic and prokaryotic cells. Acha ji. And then the membrane of nucleus is known as nuclear envelope. I believe it's not written over here, but you can just remember this fact that the membrane of nucleus is known as nuclear envelope. Then we have Golgi body. Okay, so Golgi body is also a membrane-bound organelle, only found in eukaryotic cells and not in prokaryotic cells. Then we have two types of ER. Okay, ठीक है. ER भी हमारे पास two types होते हैं. जैसे ribosomes, we have two types of ribosomes, 70S and 80S. Similarly, we have two types of ER, endoplasmic reticulum. One of them is a smooth endoplasmic reticulum. Other one is rough endoplasmic reticulum. ठीक है. So rough endoplasmic reticulum is literally attached with the nucleus, and it joins with the nucleus and extends towards the outside of the cell. And rough, we call it rough because उसकी rough appearance होती है. And why is that so? Because rough endoplasmic reticulum, they have ribosomes attached all over it. And what were ribosomes? They were the dotted structures. Okay, so dotted structures of ribosomes are basically giving a rough appearance to rough endoplasmic reticulum. I'm sure that you do not know the function of any of these, and we'll talk about it. So do not need to worry. Just try to understand what I'm trying to convey. Okay, and then the smooth endoplasmic reticulum, the name obviously specifies that it has appearance which looks smooth and not rough like RER. Okay, so smooth endoplasmic reticulum, they do not have ribosomes attached, while rough endoplasmic reticulum, it continues with the nucleus and it has ribosomes attached all over it. Make sense? Are we all clear so far? Okay. Then we'll talk about this in later class. Okay, how will we identify these? How are we going to identify Golgi? How are we going to identify RER and SER? क्योंकि इनके labelings आती हैं and you need to know their clear description. So we'll talk about everything in detail because this was just the initial orientation class today. But I need to finish the labelings of eukaryotic cell. Achha, then we have vacuoles, right? We have small vacuoles in animal cell, and we have a large permanent vacuole in plant cell. Then we have centrioles, cell membrane. We do not have a cell wall in animal cell, but we have a cell wall in plant cell and in fungi. And quick check, what is the component of plant cell wall? Usman, you can type it or you can unmute yourself.
Peptidoglycan?
No, I'm talking about the cell wall of plant cell. कोई बात नहीं. It's cellulose. Okay, cellulose. And what component do we have for the cell wall of fungi? Abdul Rahman.
Chitin?
Chitin, perfect, exactly. So we said that the cell wall of bacterial cell is made up of peptidoglycan, and the cell wall of plant cell is made up of cellulose, and the cell wall of fungi is made up of chitin. Okay, we need to remember these three terminologies because they will definitely and surely they'll come into your paper. Because when I was doing my AS level paper, this was the first question. So this is why I'm emphasizing again and again on the differences between prokaryotes and eukaryotes, and all the things that I have mentioned so far. Acha ji. Then we have ribosomes. We are aware of the fact by now that there are two types of ribosomes, 70S and 80S. 70S ribosomes are found in Hira. Quickly, 70S ribosomes are found in prokaryotes. Perfect. And 80S ribosomes are found in Iman. Iman, right here. Iman, I think she has left. So 80S ribosomes are found in eukaryotic cells. Okay, so these were the labelings that we need to memorize these labelings, but not right now. We'll discuss how to identify and how to differentiate between each one of them, because when they'll appear in your paper, we tend to make mistakes and we tend to confuse all of them. So this is it for today. We'll have our other class tomorrow at the same time, 5:15 p.m. And till then, take care. And I hope this was a brief intro of cells. We'll discuss cells again tomorrow, and we'll send you the recording in a while. All right. So Jazakallah khair and take care. Jazakallah khair for joining. Okay. Allah Hafiz.